This is the inheritance section of the A-level biology course. We're going to be looking at epistasis. As always, there's a little activity to get started. All you have to do is match up the type of inheritance with the relevant description. If you are unfamiliar with any of these types of inheritance, or you're unfamiliar with any of the terminology that's being used in this presentation, I'd suggest you go back and check out monohybrid and dihybrid inheritance presentations, which goes through some of the terminology you're going to need to pass this course at A level. What you want to do is pause the video now, take some time to try and match up each of the statements, and then check your answers. So monohybrid inheritance is simply where we have one gene. You can have as many alleles as you want, but it's only one gene linked to one characteristic or one polypeptide. Dihybrid, di meaning two, means that we have two genes, and remember we're talking about autosomal genes, which in humans is pairs number 1 to 22. In codominance, where we have different alleles of genes blending together, so in this case we've got yellow and blue producing green offspring. We've got sex-linked inheritance, which specifically is linked to chromosome pair 23 in humans, but it's the sex chromosomes. Again, it'll be a different number for different organisms. And lastly, we have the autosomal linked inheritance, which is any kind of linkage in chromosomes 1 to 22, but the characteristics of genes are on a specific chromosome. So in dihybrid, you have two genes, but they're on different chromosomes and are independent of each other, whereas in autosomal linkage, we have two genes, but they are on the same chromosome. So what actually is epistasis? Well, epistasis is a similar type setup to dihybrid inheritance in the fact that you will have two genes. However, unlike the hybrid inheritance, which will affect two different characteristics or two different polypeptides, this particular process is on the same biological pathway, so you will be talking about the same characteristic. So for example, normally it was linked in with the color or physical appearance of flowers, but it can be other things in terms of seeds, and you can of course bring colors of insects and other organisms into it, but most of the time it will focus around colors. It's the easiest thing to see in the pathway. There are many different types of epistasis. Recessive and dominant are the most common ones, but there are lots of other examples. You will not be expected to identify any of these in the exam. On one of the A-level specifications, it actually specifically says to ignore the word dominant and recessive. It simply wanted you to identify the process as epistasis. I would suggest you're familiar with ratios, especially 9:3:4. That seems to be the most common one, but you can also end up with a ratio of 12:3:1. And we'll look at some specific examples later on, and you can see what we mean in terms of ratios and outcomes. When it comes to questions on epistasis, this is a general diagram. It always suggests that you draw in the margin or somewhere around your test paper and extract information from the question and build it up. You won't always get exactly that diagram, but this is a good starting point for the moment. And what you're looking for is you need to identify which gene in the question represents gene A and which gene represents gene B. Gene A will allow you the phenotype to change from number 1 to number 2. Now in the question, it could be a homozygous dominant genotype, a heterozygous genotype, or the homozygous recessive, depending on whether the dominant or recessive allele causes the change from phenotype 1 to phenotype 2. In terms of gene B, it would be exactly the same setup. It could be either the homozygous dominant, the heterozygous, or the homozygous recessive combination. That'll let you progress from 2 to 3. It'll completely depend on the example they give you as to what will happen with A and B, but A and B are completely independent of each other. Just because the dominant allele in A progressed from phenotype 1 to 2, don't assume the dominant allele in B progresses from phenotype 2 to phenotype 3, you need to extract the information. It is important to have a diagram like this on any epistasis question in the exam, but it's also worth noting that you won't necessarily be able to get this completed. When the exam information is given to you, you may find that you can't work out which order certain phenotypes appear on, so which is number 2 and which is number 3. You may not be able to identify that if you need the dominant allele or if you need the homozygous recessive genotype to progress from phenotype 2 to 3. It doesn't really matter. You should extract as much information as you can, and there has to be enough there for you to answer the question. So don't worry if you don't get a perfect diagram, but you have to modify it in some way to fit in with the question. That's perfectly normal practice. So let's have a look at some specific examples. In this particular question, in the table in the top right of the screen, you can see that we have two genes, T and F. The question information in the top left tells us that each of those genes has a dominant and recessive version, so a dominant and recessive allele, and it gives us some phenotypes. And then the question simply asks us is, what is the phenotype of the following genotypes? I want to stress this point again because it's one of the most common mistakes that students make when dealing with questions in epistasis. Don't make assumptions in terms of phenotypes being produced with certain alleles, so don't assume that a dominant allele will progress you from phenotype 1 to 2 or 2 to 3. Sometimes it will be dominant and sometimes you will actually have a recessive condition, and you need to read the question very carefully to make sure that you're getting it right for each individual question. A good pointer as well is when it starts giving phenotypes, normally the most common type of phenotype is always going to be phenotype 1 on your diagram. I say always because it's not 100% guaranteed, but as a general rule of thumb, it's a good starting point. So if we have a look at the diagram on the top right, we can see that the most common phenotype is orange. So we're going to work on the assumption at the minute that that's phenotype 1. We can also see that in the orange, they have both got a homozygous recessive for gene F, and they've also got variations in their genotype for gene T, with one of them being heterozygous and the other one being homozygous recessive. What that suggests is that to progress along from phenotype 1 to phenotype 2, it is most likely the F gene that is causing that change. This is further backed up by the changes in their letter T's, gene T's, because changing gene T has no impact on it in the same way that if you have gene B, if you don't progress from phenotype 1 to phenotype 2, gene B becomes completely irrelevant because no matter what one in terms of alleles you've got, you haven't progressed from phenotype 1 to phenotype 2, so you cannot progress from phenotype 2 to phenotype 3. So that is leading to the suggestion that on our diagram, gene A should be replaced with the gene F and gene B should be gene T. So how do we know which gene is which on the diagram, and we know the starting phenotype as best as the information lets us work out at this point, because we may need to make changes later on. We now need to determine which version, the dominant allele or the homozygous recessive genotype, determines the direction of change from phenotype 1 to phenotype 2. Well, if we look up in the top right again at the different phenotypes, both the blue phenotype and the pink phenotype have a dominant allele for F. That would indicate that they're progressing on to phenotype 2. You need to have that dominant F. And again, by not having that, you can see both of the orange phenotypes have the homozygous recessive condition. So add that onto your diagram and then have a look at gene T. So if we now have a look at gene T, we can see that the pink phenotype is linked to the capital dominant allele for T, and that the blue phenotype occurs when we have the homozygous recessive genotype for T. So what we'd suggest is that to progress from 2 to 3, we would have one of those conditions. But it's not actually possible in this question with the information we've been given to determine if phenotype 2 is blue or if phenotype 2 is pink. We just know that if you have a dominant allele for T, then you're going to be pink. If you have a homozygous recessive genotype for T, then you're going to be blue. That's as far as the information lets us go in this question. So all I've done to my diagram is simply add in the letters for capital T and the lowercase t genotype and linked it up to the correct color. It could be that later on in the question it gives me more information and I'm able to identify the particular sequence if blue or pink comes first in the sequence. But at this point it doesn't really matter. I can answer the question with the information I've got, even if I've got the wrong order. So now that we've completed the question, good practice is to go back to the original table with the phenotypes and genotypes and just double check that your diagram still fits in with everything that's happened. So if we look at the two orange phenotypes straight off, they've got the F gene, they've got the homozygous recessive setup, therefore they are orange, which fits with our diagram. Blue and pink have a dominant allele for F and progress along onto the next bit of the diagram. Blue has the homozygous recessive genotype with little t's, which gives us blue, and if we've got a big T, it does indeed give us pink. So that all fits in with the genotypes and phenotypes that we have been given in this question. Now what we have to do is go back to the original question that we were asked and identify what are the phenotypes for the genotypes given. Well, first off, I'm going to look gene F. For A, it has a heterozygous for gene F, which means we progress along into phenotype 2, which is to determine if it's blue or pink. We look at the gene T. Gene T is homozygous dominant. It has one of those uppercase T's, so that organism would be pink. If we look at question B, again starting off on F because gene F is the first one in the sequence in the diagram, we can see again it has homozygous dominant, so it has got the capital F dominant allele, which means we go on to blue or pink, and it's got two little t's, which means that we progress on to blue. So example 2 is a little bit more complicated, and because of the way it's set out, we're going to take each of the paragraphs separately, and we're also going to have to adapt our epistasis diagram in order to fit in with this particular question. So the first part of the question makes it clear that we have one characteristic in the pigment of rabbit fur, and that one characteristic is controlled by two genes. And in the exam, when we have one characteristic controlled by two separate genes, that immediately should lead us to the process of epistasis. So in the second paragraph, it gives us information relating to the first of the two genes involved in the process. And in this particular one, rather than two alleles, we've got three alleles. So we're bringing in this idea of multiple alleles. And what we've got is we've got the normal dominant uppercase letter A, which in this case results in a black pigment due to an enzyme production. We also have a version which is AH. Again, we do get the pigment with that, but we only get pigmentation on the face, ears, legs, and tail because of the temperature variation. The enzyme becomes inactive. And then the third, which is the lowercase a, gives us a non-functioning allele, and as a result, we have no enzyme and no color change at all, so the organism remains white. So now that we've extracted the information from the question, we can go on and put that into our standard diagram for epistasis. Remember, when you're in an exam setting, you can simply use underlining or highlighting in the question rather than writing out the content all over again, as you can see on the screen, and use that to build up your diagram. You can build up your diagram without that underlining as you go through the question, but you are taking a risk because you won't have necessarily got that checking mechanism at the end. So I would always suggest underline something, add it to the diagram, underline something, add it to the diagram, and that way you're much less likely to make mistakes. The obvious first problem is that for gene A, we have 3 alleles, but in our diagram, we only have 2 little boxes. So we're going to have to adapt the diagram to get started. So we've modified the diagram to give us our 3 alleles for gene A. We have our homozygous recessive genotype, which is the 2 lowercase a's, which then gives us phenotype 1, which in this case is white fur. We also have the uppercase letter A, the dominant letter A, which gives us the black phenotype, and then we have the AH, which gives us what I'm referring to as the part black, which should be certain parts of the body would be black and other parts would be white. In the exam, you'd simply underline those sentences. You wouldn't have to write out face, ears, legs, and tail. You could underline it right in the margin, part black, and then when you're writing your answer, write the full thing out. Because you haven't defined what part black is, it would be very dangerous to use it as part of an answer, but for your interpretation, it's absolutely fine. One thing to note is in the genotypes, it's A and then a dash for black and AH and a dash for the part black. We can use a dash. The dash just simply refers to the fact that whatever that second allele is, it doesn't make any difference to the outcome in terms of phenotype. So it doesn't matter if that second letter, that second allele is the dominant or the recessive, we will still get the same phenotype. Now that we know what's going on with gene A, let's have a look at gene B. In the question, it tells us that allele B is the third paragraph in the question. It tells us that allele B, again dominant recessive, and the dominant version of B results in what is simply black fur, but it says large amounts of pigment making the fur black. What that means is that the AH from gene A would be masked in the phenotype if it allele B dominant, so the uppercase B, was present. The other version of B, the lowercase version of B, simply results in the fur appearing as brown. B will only have an effect if the organism has an uppercase A or an uppercase A and H allele as well. If the organism has the 2 lowercase alleles, the 2 lowercase a's, then the organism will be white and gene B just doesn't have any impact whatsoever. In order to put our information for gene B into the diagram, again, we're going to have to make a few tweaks. So in this particular case, because gene B produces 2 different variations in terms of phenotype, we're not progressing backwards along a chain. So we've just tweaked it and moved it so we've our 2 options. And again, we've used our dash to represent a second allele, but it doesn't matter which it is. If the organism has that uppercase B and a dash, or it has the homozygous recessive, we will end up with either black all over fur or brown fur. Again, it's kept on top of the green line so that we know we have to progress from A, uppercase, or uppercase A and H from gene A to have gene B come in to the phenotype. Again, just keeping that format, you can clearly see it and you don't make mistakes in the exam. So what we've looked at there is the interpretation of the information and type of setup that you get for the actual exam questions. Now, in terms of what the subsequent questions will be, if you can wrap your brain around all that interpretation, if you could produce your diagram, pretty much any question they give you as a follow-up, you're going to be able to answer pretty straightforward. One word of advice, when you see any question with the word explained in it, you can't just say what happens, you have to say why it happens. So you have to talk about the progression between phenotype 1 and 2 or phenotype 2 and 3 and how often you will only progress from phenotype 2 to 3 if you've progressed from phenotype 1 into phenotype 2. So just read the questions carefully. If you've done the interpretation diagram, you've done all the hard work and the marks are there for you. Just make sure, folks, that you're careful, because more and more recently, they have been blending different types of inheritance together. So epistasis has been tied together with autosomal linkage, and epistasis has also been tied in with sex linkage. So just watch out, they can blend different types of inheritance into the same question. Use the diagram, set your structure right, and you're very, very, very unlikely to lose the marks. So just to finish off, there are 5 different types of inheritance listed, and there are 5 different statements that connect to each one of those. All you have to do is match the ones on the left with the ones on the right. And pause the video now and give yourself a couple of minutes to have a go at that, and then you can check your answers. In monohybrid inheritance, the ratio should be a 3-to-1 ratio. Make sure, folks, that is only the phenotype ratios and only where you have 2 heterozygous parents. In terms of dihybrid, again, 2 heterozygous parents, we will have a 9:3:1 ratio. Your codominance would normally give you a 1:1 ratio. Epistasis, there's a number of possible outcomes. The one that's most likely to appear in the exam will give you a 9:3:4, but there are other versions of it, so just carefully read the question. You may need to do a genetic diagram beforehand just to make sure you get the right ratios. And then autosomal linkage is the one that remains, but in autosomal linkage, remember, we have 2 of the phenotypes will be in particularly high numbers in the population, whereas the other 2 phenotypes will be in very low numbers. And remember, those low number ones are called the recombinants. And that's the end of the epistasis presentation.